During a Red Cross relief mission to Africa in the 1960s, a Norwegian doctor saw pregnant women do something a little odd. When the time for birth arrived, they sipped a medicinal tea prepared by local witch doctors. Made from the leaves of a plant called kalata-kalata, the tea helped them go into labor and deliver more quickly. Scientists discovered that the tea worked because it contained a mysterious protein that eased pain and made the uterus contract. Scientists named it kalata-1. Almost 30 years later, David Craik made a scientific breakthrough that promises to turn kalata-like proteins, which are known as cyclotides, into new drugs for treating health problems that affect millions of people. Some of these health problems include antibiotic-resistant bacteria and even AIDS. For that work, the American Chemical Society awarded its 2011 Ralph F. Hirschman Award in Peptide Chemistry to Dr. Craik. Proteins are already some of our most important medicines. Insulin, for example, is a protein. Many other proteins could become life-saving medicines, but there's a problem with proteins and smaller chunks of proteins called peptides.
In general, peptides are regarded as great drug leads because they're very potent and very specific for their receptors, but they don't make good actual drugs because they get digested as soon as you swallow a tablet. Your body can't tell the difference between a piece of steak and a protein-based drug, and it just wants to chew both of them up the same way, and so the drug can't get to the site of action.
Craik discovered that kalata-1 has a molecular structure, or architecture, that shrugs off digestion in the stomach. Just like a brick building can withstand a storm, this protein can withstand forces that threaten to pull it apart. Other proteins, or peptides, consist of units known as amino acids, linked together in a straight line. But Craik's research revealed a protein that looked like a bicycle tire, a circular protein with the amino acids knotted together. Quite understandably, Craik named it a cyclotide, cyclo for circular and tide for peptide.
But of course the topology immediately explains why the protein's so stable. Remember, the African women can boil it in the tea and then drink it, and most proteins you can't boil. Your morning eggs denature when you heat them up, and that's basically because of protein denaturing. But this molecule can withstand the boiling and the oral ingestion, and it's that knotted structure that makes that so.
A bounty of cyclotides already exist in nature, in plants like kalata-kalata. Plants don't produce just one cyclotide, but whole suites of them, 20, 50, even 100. Craik believes that there are tens of thousands of such cyclotides in plants. And that leads us to one of the ways in which Craik's award-winning research may impact the everyday lives of millions of people.
Plants, unlike animals, if they're attacked, they can't run away, so they have to have a very good chemical armoury to protect themselves from herbivores or from pathogens. So cyclotides turn out to be defence molecules. They're very potent insecticidal agents. So one potential application would be to take the genes that encode for cyclotides and put them into plants that are attacked by insects but don't have cyclotides, and so we could basically provide a natural insecticide. And so cotton would be an obvious application there because it's attacked by insect pests.
Plants genetically engineered to manufacture their own insecticides could mean more food for a world with more than 1 billion hungry people. But the main application of knowledge about cyclotides may be in medicine. Scientists could take the cyclotide structure, re-engineer it, and graft it onto proteins, turning them into drugs that could be taken orally.
Well, cyclotides have the ability to combat the virus that causes AIDS. We don't know the mechanism, but we think it might be by inhibiting the replication process, but that's work that's ongoing at the moment. They're also active against microbes, so and this may be related to a natural function in plants as well, to basically kill pathogens that attack plants. They also are active against a number of human pathogens, including Staph aureus.